So our next speaker is Paul Grossfeld. He's a professor of pediatric cardiology at the University of California in San Diego. He's going to va a hablar sobre los factores genéticos relacionados con la predisposición a desarrollar problemas neurocognitivos y de conducta.
Gracias. Voy a decir un poquito cosas en español primero y entonces voy a cambiar a inglés. Can you change so I have the presenter's version of the PowerPoint? Primero yo quiero decir gracias a todos de ustedes por invitarme. Es un grande privilegio para estar aquí. So, gracias. Y también yo quiero decir gracias a Jimena y Cosme para preparando esta conferencia. Yo sé de mi experiencia que es muy difícil, necesita mucho trabajo para preparar una conferencia como esta. So, gracias. Y la cosa última es, yo quiero decir, lo siento para mi español muy malo, y por eso ahorita voy a cambiar a inglés. During the talk, if anybody has questions, please stop and ask. Okay. Can we change so I can see the presenter's notes or no? No? All right, I guess I have to remember everything I put in my slides. So I was asked to give a talk about the cognitive and behavioral problems in Jacobsen syndrome. And first I want to say I am a pediatric cardiologist. I am not a neuropsychologist or neurologist. So please forgive me because this is really not my area of expertise. Do I advance the slides or? I guess they do. Teresa, how did you advance the slides? Let's see. There we go. Okay, good. So this is a picture from a few years ago at one of our conferences in San Diego. And all four of these children have the same very severe heart defect called hypoplastic left heart syndrome. But even with that very severe heart defect, you can see that there are very big differences in their level of functioning and their behavior. So as we became interested in this area, there were some very important questions that we need to ask. So I'm just going to read these. The first one is, are there genes in 11q that are involved in brain development and function? And if so, is there a correlation between the presence or absence of any of these genes and the level of cognitive ability or their behavior? And then once we identify possible genes, can we use animal models to study the role of these genes for human problems? And then later today, there will be a second talk on possible treatments or therapies based on what we've learned so far about these genes. So first, like Dr. Martina described, it's very important to determine exactly the deletion breakpoint. And then we have to perform very comprehensive assessments of the children. And from that, we can identify potential candidate genes based on their presence or absence and their known functions. And with that information, we then use animal models for understanding the mechanisms of these genes. And I'll show you a little bit about those studies today. In the future, we will actually be studying cells from patients with Jacobsen syndrome. If we have time, I'll tell you a little more about those studies later. And the ultimate goal is to develop potential treatment strategies. Does this make sense so far? All right, so first I'm going to talk about what we know about the cognitive function, and then some of the behavioral features, And along the way, I will describe the candidate genes. And then, as I said, later today, I'll have a separate short talk about possible clinical trials and drug development. Okay, and the kinds of tests that we use for cognitive function are very standardized tests. All of these studies were done in English-speaking patients, but I'm sure most of these studies can be done in other languages too, like Spanish. And this is just a list of some of these studies. I won't go through all the names. Then there are other studies for behavior. There's a very standardized test for autism called ADOS, which stands for Autism Diagnostic Observation Schedule. And I should also say all of these tests are specific for the age of the child. And then there are a number of tests that are questionnaires that assess for behavior. So these are questionnaires that are done by the parents usually. And there are always limitations to questionnaires, biases, but overall we have found these tests to be quite helpful. And these are just a few more of those tests for different age groups. Okay, so here I want to summarize some of the results of the cognitive testing. So if we talk about IQ or full-scale IQ, there is a very wide range of IQ scores. So you can go from a child with low normal IQ of about 80 to very severe impairment. And as you can imagine, there is a very strong correlation between the size of the deletion and the severity of the cognitive impairment. And we have identified so far at least 4 regions in chromosome 11 that we think contain genes that contribute to these problems. And I'll show you on the next slide one region that we think contains a gene that's critical for brain function, because we know that every child who has a large deletion with this gene has very significant cognitive impairment. So I'm sorry, I know this is a very complicated slide, so I want to show you a few things about this slide. So this shows the end of chromosome 11. That's the end of the chromosome called the telomere. And what we see is that if even children with very small deletions, not really with Jacobsen syndrome, but they can have mild cognitive dysfunction. And so we think there's probably at least one gene in this region. And if you go all the way out over here to the children that have the largest deletion, they are the ones with the most severe impairment. And specifically, that means anybody with a deletion of more than 12 megabases. And then there are children with Jacobsen syndrome that have small deletions up to about here. And they tend to be the most mildly affected. And then there are children whose deletion goes up to about here. They are relatively mildly affected as well, but maybe a little more affected than those with the small deletions. And again, if you have a little bit larger deletion to include this region, those children without exception have the most significant impairment. And there are some children that have just deletions in these small regions, what we call interstitial deletions. And if it's just affecting one of these regions, then they are mildly affected. If it's an interstitial deletion, let's say, that involves this region and this region, or all 3 of these regions, then they also are very severely affected. And when I say severely affected, that means IQ of less than 50. And if it's mildly affected, then IQ greater than 60. Does this make sense? Too confusing?
Just one thing, like most of us know the case of the deletion.
Yes.
24 point something, 23 point something.
What does that mean?
We don't know those numbers.
So that's a very, very important question. I'll repeat the question. Jimena asked, what does it mean if they say your child's deletion is 24.1, let's say? That's not very helpful because, like Teresa showed, we need much more detailed information to know exactly which genes are affected. And that's when you get a number that says something like 10 megabases from the array study. And that gives us very important information because it gives us a readout of exactly which genes are affected.
So over that, like the
So this would be 23.3, 24.1, 24.2, 24.3. Not very helpful for genes. The key is to know which genes are affected. So in the future, we never want to talk about 23.3 or 24.1. The information we need is from the array study, like megabases. Does that make sense? Any other questions? Okay. Let me go back. One other thing that's very important about these studies, just to repeat, so far without any exception, and that means probably over 30 or 40 patients now, any child with a deletion of at least 12 megabases has the most severe cognitive impairment. And without exception, any child that has an IQ of greater than 60 has a smaller deletion. So that's helpful to try to predict the future of how your children might do, especially for a new baby. But sometimes, even a child with a small deletion may be more severely affected because there could be other genes from other chromosomes that might affect that. Does that make sense? So this is another way just to show that if you have a very large deletion over here, the IQ is lower. Compared to a medium or a smaller deletion, the IQ is much higher. And this difference between the large deletion and a slightly smaller deletion suggests there's a gene, maybe, that's responsible, like I said. And this just summarizes what I already said. So, greater than 12 megabase, more severe impairment. And anybody who tests higher has deletions smaller than 12 megabases. Then what we wanted to do is look in this region that distinguishes the bigger deletion from the smaller deletion. And it turns out there's one gene in that region that we think is probably the cause of why children with larger deletions are more severely affected. And the name of this gene actually has the word brain in it. So that's a pretty good clue that this gene might be very important. And this gene is called BSX1, or brain homeobox protein. And as far as I know, there's only one researcher in the whole world that's studying this gene. And he is in Germany, in Berlin, Dr. Trier. And so far, he has shown that this gene regulates appetite and locomotory function, and it's only expressed in a small group of cells in the brain. But based on what we think, it suggests this gene may have other more important functions for cognitive function. And Dr. Trier is now studying mice where we can specifically just eliminate that one gene. He hasn't published this yet, but he tells me that when you eliminate this gene in mice, he can do tests to show that these mice have very significant cognitive impairment. Does that make sense? Nobody is sleeping yet? Okay. So that's the gene that we think is responsible for the very significant impairment. And I told you there is even a small difference between people that have a medium deletion, a medium deletion and a small deletion for something called digit span. It's a measure of short-term memory. So if I give you 5 words and ask you to tell me those 5 words back 2 minutes later, children that have a medium or larger deletion have a much more difficult time doing that than those with small deletion. And similarly, we have found a gene that we think is only deleted in these patients, but not in these patients, that may be the cause of this difference. And this is a gene called neurogranin. And in mice, the more neurogranin there is in the brain, the stronger there is a signal for learning and memory. What's also very interesting is that neurogranin is probably important for helping environmental enrichment, which I'll talk about. And this could have very important implications for other patients that have learning problems and memory problems. For example, patients with Alzheimer's disease or some patients that have suffered strokes. Because as some of you may know, one of the ways that we try to treat patients with these problems is from environmental enrichment and stimulation. So here's what we know about what neurogranin does. It helps the ability to make memories, to learn and remember things, and it helps long-term memory. So similarly, there are mice that have been made that are missing this gene, and I won't go through all this, but we know that mice missing this gene have learning and memory problems. And interestingly, they appear to be very anxious and have decreased attention. And that might affect their ability to learn as well. Now, if you place these mice that are missing the neurogranin gene, or even a normal mouse, in what we call a rich environment. So, for example, you give them toys, you give them an exercise wheel, and you give them friends to play with, other mice. If you keep giving them this enrichment protocol, they can improve their memory and learning function. This is a very important slide that I want to try to explain to you. So, this on the left is the learning or IQ of a mouse that has not been exposed to the environmental enrichment. And the black is what happens to the mouse's learning when they've been exposed to environmental enrichment. Now, just like humans, mice have 2 copies of every gene, like for neurogranin. And if we make a mouse that is missing both copies of that neurogranin gene, when we expose them to environmental enrichment, there's no improvement. However, if we take a mouse that is missing 1 copy of the neurogranin gene, like some of your children, and then if we put them in that environmental enrichment, they improve their learning almost to what a normal mouse can do without environmental enrichment. So this change, I think, could be very important for your children. What's also very important for the rest of us is even a normal mouse with 2 copies of neurogranin improves their function as well when placed in environmental enrichment. That's a very complicated slide, but does that make sense? So we think this might be an opportunity to help children with their memory and learning problems if we can improve the neurogranin level in their brains. And maybe other people as well, not just children with Jacobsen syndrome. As an example, as I said, it turns out some patients with Alzheimer's disease have been found to have decreased neurogranin protein in their brains. So maybe even in patients with Alzheimer's disease, if we can boost up the level of the neurogranin protein in their brain, that could help them too. And I can tell you from my own experience with my father, it would be too late, but in the future, it would be nice to have some therapies for those patients, because right now there are no good therapies. So this is the real question that we want to ask. So can increasing neurogranin improve memory and learning?
So he's asking where is this gene located?
This is on chromosome 11, and it's about 10 megabases up from the end of the chromosome. So if your child has had the array to find exactly where the deletion is, you should be able to look at the report and see if neurogranin is affected by the deletion. That's why that information is so important. Okay, so that's the story for cognition so far. So I'm going to change subjects to talk a little bit about autism. And I must say that I was very shocked when I first learned in about 2004 that this child was actually diagnosed with autism. And she is now about, I think, how old is she? Maybe 16 years old now. So there were a few other cases since 2004 of children with Jacobsen syndrome and autism. So as much as I couldn't believe those results, I thought we need to really figure this out if it's true. And so with my colleagues in San Diego, in the last few conferences, we studied 17 patients. There were 5 males and 12 females. We found that 4 out of the 5 males satisfy the diagnosis of autism or autism spectrum disorder, and about 1/3 of the females, 4 out of 12. So overall, about half of your children would fulfill the diagnosis of autism. So with that information, we wanted to find out if there might be a gene that's responsible. And what we found is there was no correlation between the deletion size and whether or not your child has autism. So the smallest deletion size was just under 9 megabases. And so this is kind of like what I showed you before. This is a child with autism with the smallest deletion to the end. These are 2 other children with autism that have what we call the interstitial deletion. And you can see there's a small region of overlap, which made us think there could be a gene in this region right here. And then we came across a child with some intellectual disability from England who was found to have a very tiny deletion in this same region of overlap. And we went back and tested that child and determined that she actually has some features of autism as well. So that made us think that there is probably at least one gene in this very tiny region of the overlap that can cause autism. And it turns out, again, this region of overlap has only about 2 genes. And one of them is a very interesting gene that's involved in brain development. And the name of this gene is just an abbreviation, RIX, or this name. And this is a very complicated description of what this gene does. But think of it as a protein that's critical to help the brain form the right connections. And it turns out one of the major theories about autism is that during the development of the brain, the normal connections do not form. It is not from vaccines, I promise you. So it turns out that there is only one group in the world that I know of that's been studying this gene, and this group is located in Tokyo. And so I was very lucky to meet with this group of researchers in Tokyo about 2 years ago. They were studying this gene to understand its role in brain development, and they had already shown that mice that are missing this gene have some cognitive dysfunction, but they never suspected that they could have features of autism as well. So when I met with them 2 years ago, I asked them 2 very, very important questions. I begged them, I said, please, can you go back and look at your mice to see if they might have autistic behaviors? And the second question I asked them is, based on all the elegant work that they have done about this gene, could we come up with a way to treat these mice and potentially your children? So now I want to show you some of the data that they have now obtained since that meeting we had about 2 years ago. So on the left, you're going to see a mouse that has both copies of this gene. And on the right, this is a mouse that is missing both copies of this gene. And I think you'll appreciate the difference. Let's see if this works. Okay, so this is a test for one feature of autism called marble bearing. And just keep watching for a few minutes. I think you'll appreciate the difference. You can see the mouse on the left is pretty happy. Sometimes he stands up and looks around to see what's going on. And overall, he's not doing too much with these marbles. The mouse on the right sometimes looks up but spends a lot of time doing this repetitive digging behavior. Somebody said this looks like people that do research on Jacobsen syndrome too. We can actually quantify these differences. So this is the mouse on the left, before and after. So a little bit, but not that different. And then the mouse that's missing this gene has a pretty dramatic difference. And even when it's just missing one gene, one copy of the gene, like for most of your children, the mice showed the same big difference. So this was indicating that, at least for this type of behavior, the mice had some autistic features. We can also do other kinds of social behavior assessments, like smelling something that's put on a Q-tip. And we know that if you give them food, they can smell fine. But in the normal mouse, shown here in blue, they will become interested in the smell of another mouse. But if the mice are missing one or two copies of the gene, they are much less interested in the smell of other mice. And that's another very important feature of autism, is decreased social interaction. And this is a very, very busy slide, which I'll talk about later. But this shows all the work done by the group in Tokyo. And I'll go through this later today in a little more detail to show you that there might be a therapy to treat your children that have autistic features, based on what they have learned about what this gene does in the brain. So, in a more general sense, could there be any treatments for your children with intellectual disability? And I will talk about that again a little later on today. And lastly, I just want to talk about ADHD. And again, there are different types of questionnaires that were used, but overall it turns out about half of your children have the diagnosis of ADHD. And other behavior problems are much, much less frequent. And this is just another way of showing for ADHD, about 1/3 don't have problems, about 1/3 are kind of borderline, and almost a half or so have the diagnosis. And very different for other kinds of behavioral problems. How am I doing for time? Done?
Almost.
Okay. I'll just go through this very quickly
possible, yeah.
in 2 minutes. Just to show now, there is some very, very exciting new technology that allows us to study your children, in ways that never would have been possible just 5 years ago, thanks to these two great researchers who were awarded the Nobel Prize a few years ago. And what they showed is that you can take, let's say, skin cells or blood cells from a patient, any patient, and you can reprogram those cells to become stem cells, and then we can reprogram the cells to become brain cells, heart cells, or potentially any other cell that you're interested in studying. So, for example, we could figure out, without doing a brain biopsy, maybe if there are differences in brain cells that are derived from your children compared to other children. This is an example of what we can show for heart cells. So these are actual heart cells that are beating just like a heart from a normal patient, from those stem cells from the skin cells. And similarly, these are actually brain cells, and they have a green label. If you watch very carefully, you'll see little green flashes. Those are actually the brain cells in a petri dish that are functioning. And that gives us the opportunity to learn what's different about your children compared to other children. And it might even give us a very powerful opportunity to test new drugs that could improve the problem before you ever treat a patient. And we've now started these kinds of studies in some children with Jacobsen syndrome. All right, so let me just summarize. This is old news, I'm sure, to most of you, and that is children or people with Jacobsen syndrome have a wide range of cognitive impairment. The BSX and neurogranin genes seem to be very important. Deletion of the BSX gene with one or more of the other genes causes the most severe cognitive impairment, with no exceptions so far. Even people with the smallest deletions usually have maybe some mild cognitive impairment, but some of those can do very well, as you know in this room. We have identified now about half of the people have autism, and we've identified at least one gene that we think is the cause. There will probably be more than one gene in 11q that can cause autism. Similarly, about half of your children have ADHD. So overall, we know that there are even for all your children that supposedly have the same syndrome. But at the same time, we think there are some very exciting potential opportunities for some clinical trials, and even maybe for the development of new drugs. And I want to thank many people from around the world that have been critical to help me to be able to tell you everything I've just told you in the last 45 minutes. And I have two psychology researchers in San Diego, Dr. Aksumoff and Dr. Mattson, Dr. Trier in Berlin, who's done this work on the BSX gene, Dr. Huang and his colleagues at the National Institutes of Health, who has done the very elegant work on neurogranin, Dr. Akiyama and his colleagues in Tokyo, who have been so gracious to work with me on the autism story, and Dr. Chugani, whose work I'll tell you about a little later today, that we are very excited about, that can form the basis for a clinical trial, which we think we can start very soon if we can get a little more funding. And finally, I want to take this opportunity to invite all of you to come to our next conference in San Diego. And I think you'll have a great time there, and we do speak better Spanish than I do. Thank you very much for your attention.